Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize winning lab. This channel has three main goals. One, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. Two, to build a supportive community where we can exchange the most valuable experience based research advice. Three, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the third goal. Let's go! Let's make great science together.
Welcome to the deep dive. Okay, imagine this. What if a cell's shape, before it splits, isn't just random?
Right. What if it's actually telling the cell what to do?
Exactly. Like dictating if its kids will be identical twins or totally different. We're talking about a hidden geometry code.
A code that governs cell fate. It's a really fascinating idea.
So today we're digging into this groundbreaking paper from Science. It really seems to flip the script on how we think about cell division, shape, and identity.
Yeah, how identity gets passed down, or sometimes how it's deliberately not passed down equally.
Our mission here is to unpack how that pre-division shape acts as this, instruction manual.
Revealing this really sophisticated link between the physical form, what's moving around inside the cell, and ultimately what the new cells become.
So let's start with the basics. For ages, the textbook picture is pretty clear, right? Cells getting ready to divide sort of bunch up.
They round up into a sphere. Mitotic rounding.
Which makes intuitive sense. You want to package everything up neatly, make sure the DNA, the organelles, everything gets split evenly.
Right. It's designed for a clean, symmetrical split, usually giving you two identical daughter cells, same size, same stuff inside, high-fidelity copying.
But that's not the whole story.
Well, that's the classical view, and it is super important for many cell divisions. But developmental biologists, the ones watching tissues actually form,
They see something different.
they've seen for a long time that cells in developing tissues often divide in very specific ways, oriented just so. It hints that maybe rounding isn't the only way or always the goal.
Okay.
What wasn't clear, though, was whether the cell's shape before mitosis could be the instruction itself. Could it tell the cell how to divide symmetrically or asymmetrically?
And how to actually dole out the important molecules, the ones that decide what a cell becomes.
Exactly, those fate determinants. This paper tackles that specific question. It takes cell shape from just being, correlated with the outcome to being potentially an instructive input, a programmable thing.
Fascinating stuff. Okay, so let's get into what they actually found. What did the research show about how cells divide in a real living tissue?
Yeah, this is where it gets really interesting. They use some pretty advanced tools.
Like what?
Things like single-cell morphometrics, basically, super precise ways to measure the exact shape of individual cells and live imaging.
So watching it happen in real time.
In real time, inside living zebrafish embryos, looking specifically at developing blood vessels and these things called neural crest cells.
Oh yeah, those are the ones that move around a lot during development.
That's right, super important migratory cells. And what they saw was, well, pretty remarkable, a way of dividing that hadn't really been characterized before. They called it isomorphic division.
Isomorphic. That sounds
Yeah.
counterintuitive for division. Iso means same, morphic means shape. Same shape division.
Kind of. That's the surprising part. Unlike the classic rounding up,
Right.
in this isomorphic division, certain cells, these motile, sort of stretchy, mesenchymal-like cells, they just don't round up.
They don't.
Nope. They actually keep their pre-mitotic shape all the way through the division process. It's kind of amazing to see.
So if a cell is long and thin, it just stays long and thin while it splits.
Pretty much. Instead of becoming a little ball, it maintains that elongated form.
Wow. So the starting shape isn't just incidental then. It's actually doing something.
It seems to be directly instructive. They found this really strong link. The cell's shape before division, its interphase morphology, especially how elongated it was, that consistently predicted whether it would do this isomorphic division. And here's the kicker.
Okay.
The highly elongated cells that divided isomorphically, they almost always produced asymmetric daughters.
Meaning different sizes.
Different sizes, yes. But the less elongated cells, the ones that did round up in the traditional way, they produced daughters of equal size, symmetrical.
So there's a direct correlation. Shape predicts symmetry, like a code.
Exactly, a shape code. It suggests the cell's physical form might pre-program the outcome, not just the genes.
And was this just like a weird zebrafish blood vessel thing, or did they see it elsewhere?
No, that's a key point.
Yeah.
They saw this conserved across different types of these motile mesenchymal-like cells in vivo,
Including those neural crest cells you mentioned.
including the trunk neural crest cells, yes, which really strengthens the idea that this isn't some fluke. It's a general mechanism.
So the cell's shape before dividing seems to tune how it divides the mode, the symmetry.
Precisely. Linking the forces shaping the tissue directly to what happens inside individual cells.
Okay, but that raises a question. Why do cells in a standard lab dish usually round up nicely, but in a living organism, they might do this isomorphic thing? The environment must matter, right?
Hugely. The environment is critical. They tested this directly.
Oh.
They took human endothelial cells, the ones lining our blood vessels, and put them not on a flat dish, but in more complex, confined environments, sort of mimicking a real tissue.
Like squeezing them a bit.
Kind of, yeah. They called them 2.5D or 3D microenvironments. And what happened? The cells consistently switched.
Switch to isomorphic division.
Yes, they shifted to isomorphic and asymmetric divisions more often. Just like in the zebrafish, it suggests the physical constraints of a tissue naturally challenge that rounding process.
Okay, so if the environment can push them towards this, could they deliberately force it in the lab to really prove cause and effect?
They could, and they did. This was a really neat part of the study. They used tools called micropatterns.
What do those do?
They let you basically print shapes onto a surface, forcing cells to grow into a specific geometry.
Constraining their shape.
Exactly. So they constrain the shape, and then they also use drugs that mess with the cell's ability to round up.
Like throwing a wrench in the works of mitotic rounding.
Precisely. Things like ROCK inhibitors, which affect the cell's internal muscles. And when they did that
Let me guess, they got asymmetric divisions.
robustly, they could reliably switch cells from the default symmetric division to this asymmetric isomorphic division. It showed a direct causal link.
That's pretty convincing. So if you stop the rounding, the shape it's stuck in becomes the deciding factor.
That's exactly what they showed. If rounding was blocked and the cell started in, say, a polarized arm shape with the nucleus off to one side,
Yeah.
it reliably divided asymmetrically unequal daughters. But if it started in a more symmetric box shape, even if you blocked rounding,
It's so divided symmetrically.
it still produced symmetric daughters. So it's not just preventing rounding, it's about the specific interphase shape the cell has when it's forced not to round. That shape tunes the symmetry.
So what specific features of that starting shape make up this geometric code? Is it just how long it is, or?
They drilled down into that. Their detailed measurements pointed to two key things.
Okay.
First, cell length or elongation, just how stretched out it was. Cells with longer bits sticking out.
Protrusions.
Yeah, protrusions. Those cells showed less rounding and way more asymmetry in daughter cell size, especially if rounding was already a bit inhibited. So elongation seems to be a major trigger.
And the second thing,
The position of the nucleus within the cell.
the nucleus position. Really? That specific?
It seems so. They found that where the nucleus was located just before division started in pre-prophase was a very strong predictor of where the division machinery would set up.
Where the chromosomes line up and where the cell splits.
Exactly, the metaphase plate and the cleavage furrow. If the nucleus was off-center, polarized.
The division was asymmetric.
Consistently, they saw this in their engineered cells and, importantly, confirmed it in those living zebrafish endothelial cells, the TECs.
So the nucleus position helps determine the cut.
It seems to be a crucial part of the code, yes. How the cell controls where that division plane forms.
Wow. So putting it all together,
Mm-hmm.
an elongated shape, an asymmetric shape, plus a specifically positioned nucleus.
That combination acts as this geometric code. It defines the mode of division rounding or isomorphic and the symmetry equal or unequal daughters.
It's like a physical blueprint set before division even kicks off.
Right. A pre-encoded physical variable that dictates a specific outcome. It's moving beyond just thinking about genes and signals. The physical structure itself is instructive.
Okay, this is incredibly cool for understanding cell division mechanics. But why should, you listening care? What's the bigger picture significance here?
The so what question. It's critical. Because this isomorphic division isn't just about making different size cells
It affects what they become.
directly.
Mhm.
It drives the asymmetric inheritance of key molecules, fate determinants. signaling molecules.
The things that give a cell its identity and tell it what job to do.
Exactly. These get unequally split between the daughters during isomorphic division. And that difference is fundamental for building tissues, for development, maybe even for things going wrong in disease.
Can you give us a specific example? How does this shape-driven asymmetry actually change cell fate?
Sure. They looked closely at a receptor called VEGFR2. It's really important for blood vessel growth. In cells doing this isomorphic division, if the VEGFR2 was already sort of polarized, unevenly distributed before division, it got robustly partitioned asymmetrically to the daughters. One daughter got more, the other got less.
And that matters because?
Because VEGFR2 receives signals. So the daughter cells ended up with different levels of downstream signaling, like this molecule PERK.
Which is like an internal do this command.
Basically, yes. And those different signal levels translated into different identities. Like one cell might become a leader cell, spearheading new vessel growth.
Well, the other becomes a follower.
Exactly. Different roles based on inheriting different amounts of that key receptor all trace back to the parent cell shape.
That's fascinating. Was everything split unevenly?
Interestingly, no. They looked at another important molecule, DLL4, also involved in vessel development. DLL4 was always partitioned symmetrically, regardless of the division mode or shape.
Ah, so the geometric code is specific. It doesn't just randomly, unevenly distribute everything.
Right. It highlights the specificity of the mechanism. Some determinants are sensitive to this geometric control, others aren't.
So how do the cells physically do this? How do they avoid rounding up if that's the typical default? What's going on inside?
Yeah, the paper digs into the mechanics. It comes down to the cytoskeleton.
The cell's internal skeleton and muscles.
Pretty much. Specifically, the actin filaments and myosin motors right under the cell surface, the cortex. These are what normally drive that rounding contraction.
Okay.
They found that in these elongated endothelial cells, when they divided isomorphically, the cell basically failed to pull back its long extensions.
The protrusions.
Right. It kept its filopodia, those little feelers, active during mitosis, so it actively resisted or evaded full rounding.
And could they mess with that? Could they force them to round?
They could, by subtly tweaking the molecules that control that actin-myosin machinery.
The actin-myosin modulators?
Yes. Even small changes could force those elongated cells to actually round up. And when they did,
They switch back to symmetric divisions.
exactly. Rounding led to symmetric division, and importantly, it also wiped out the asymmetric partitioning of VEGFR2. It's all linked: the shape, the mechanics, the fate determinant distribution.
Okay, we have the geometric code, the shape, the nucleus position. We know it leads to asymmetric inheritance of things like VEGFR2. But what's the go-between? What's actually reading the code and making sure VEGFR2 goes to the right place during this weird isomorphic division?
This is maybe one of the coolest, most innovative parts of the study. They found the messenger,
What is it?
little vesicles inside the cell called Rab4 positive recycling endosomes.
Rab4 endosomes. What do they normally do?
They're part of the cell's internal trafficking system. Think of them like little sorting hubs, crucial for recycling proteins, including receptors like VEGFR2, back to the cell surface where they can do their job.
Okay, like a cellular return-to-sender system almost.
Sort of, yeah, or bringing things back to the front door. And they found something really striking about these Rab4 endosomes.
What was that?
While other types of endosomes involved in different cargo sorting got all mixed up and divided symmetrically,
Yeah.
the Rab4 compartment didn't. It actually maintained its polarized position from before division all the way through isomorphic division.
It stayed put in its original location.
Yes, which is super unusual during mitosis, and that's critical.
Why? How does that link to VEGFR2?
Because Rab4 endosomes carry VEGFR2. By staying put, by maintaining their polarized distribution, they act like fixed delivery points.
Ensuring the VEGFR2 gets delivered asymmetrically to the daughters.
Precisely. They ensure the asymmetric delivery based on where they were positioned according to the cell's original shape code.
Wow. And could they prove that link?
They did. They silenced the Rab4 protein, so the endosomes couldn't function properly.
And even if the cell still divided isomorphically, kept its shape.
VEGFR2 was then split symmetrically. It completely broke the connection between the cell's shape and the daughter cell's fate.
That closes the loop beautifully.
It really does. It shows how the physical mechanics, the cell shape, directly talks to the trafficking machinery, the Rab4 endosomes, which then dictates the identity signal, the VEGFR2 partitioning, mechanics to trafficking to fate. It's a really elegant cascade.
This feels like more than just adding a detail. It feels like it's fundamentally changing how we should think about cell division, doesn't it?
Absolutely. I think it's a genuine breakthrough because it takes cell geometry from just being something that happens to correlate with division
Right, just an observation.
to being an instructive variable, something that actively directs the outcome. It's like discovering a new way cells can be programmed,
Yeah.
which has huge implications.
How is it innovative, specifically? What were the key steps forward here?
Well, several things. First, just developing those quantitative single-cell shape maps was a big step, really measuring morphology precisely.
Okay.
Second, obviously identifying and characterizing isomorphic division itself. That's a fundamental discovery about how cells can divide.
And third,
Perhaps the most impactful is making that mechanistic link, connecting the physical shape through the Rab4 endosomes acting as readers to the asymmetric routing of fate determinants.
unifying mechanics, trafficking, and fate.
Exactly. Into one coherent framework. That opens up so many new questions.
So where does this lead? What are the next big questions for researchers now?
Oh, plenty. For starters, how do real tissue environments, which are even more complex and constrained, influence this?
Or disease tissues, like in cancer, where the environment around cells changes drastically.
Right. Does that mess with the code? Does it scramble the signals? Could changes in tissue stiffness, for instance, alter how cells divide and differentiate via this mechanism?
And thinking ahead, could we potentially use this?
That's the exciting prospect. Could we find ways to maybe non-invasively manipulate cell shape?
To guide what stem cells become, perhaps?
Or help?
Tissues regenerate?
Potentially, yes. If shape is an instructive cue, maybe we can provide the right physical cues to get desired outcomes.
And there's still that fundamental question about rounding.
Absolutely. Mitotic rounding has always been seen as critical for making sure chromosomes divide correctly, avoiding errors.
So how do these isomorphic cells get away with not rounding? How do they maintain genome stability?
That's a huge open question. Do they have different checkpoint mechanisms? Or is rounding maybe not as universally essential as we thought? Uncovering that could teach us a lot about basic cell cycle control.
So wrapping up, what's the key takeaway for you, the listener, interested in how biology works?
I think it's that this beautiful, complex process of cell division is even more nuanced than we imagined. It's not just genetics running the show.
The physical shape, the geometry, is playing a direct, instructive role.
Exactly. It's pre-programmed by these physical cues. There's an elegant choreography between the cell's form and its function, determining fate right from the moment of division.
Okay, and for that final provocative thought to leave everyone with.
Well, just consider this. How much information about a cell's history, its current neighborhood, and its daughter's future might be encoded not just in DNA or signals, but in its actual physical shape before it divides? What other shape codes might be out there waiting for us to decipher? And if we can understand them, could we unlock entirely new ways to control cells in health, development, and maybe even in fighting disease?